Moving on, as they say, we're now going to Japan. And could I welcome Ito-san, Dr. Ito-san, and the floor is yours.
Great. Thank you for kind introduction. Right? Okay. Maybe the title page is skipped. But yeah, thank you for your attendance to my presentation. And I'm going to talk about an organochip system and its application for the toxicological assessment of our heated tobacco product. And before jump into my presentation, I'd like to mention that I will have a Q&A session at the end of my presentation, so that please hold a question until the end of my talk. This is today's outline. I'm beginning with what organochip is, and then I will introduce a case study with basket on a chip system for risk assessment of tobacco product. Yeah. So what is organochip? It certainly doesn't mean that the actual organ is placed on a chip, but cells are on a chip. And as you may know, human body is composed of 40 to 60 trillion cells, and also organs are composed of cells. So each organ has its specific cells to exhibit organ function. And cells can be isolated from organ, and researchers normally use isolated cells because it's commercially available. So researchers purchase the cells and multiply if necessary, and then use for the biological study. There are several ways for cell culture. The simplest way is 2D culture. 2D culture is that cells are sheeted on the culture dish, plastic dish in monolayer, so it's very easy to use and provide high throughput. But as you can imagine, that your organ is not flat. Your organ is 3D, so sometimes the cells under 2D culture lose their original characteristics. One of the solutions is 3D culture technique. 3D culture technique aims to recreate three-dimensional architecture by providing cells with supporting matrix or scaffold, and then the cells regain their original function. So it is considered more predictive in terms of toxicology and pharmacology, and organochip also utilizes a 3D culture system on a chip. But its different point is a reproduction of dynamism of organs or dynamism of surrounding environment of organ. Yeah. So what is dynamism? In the context of biology, dynamism is generated by various physiological processes. For example, during breathing, your diaphragm moves downward to expand lungs, and also the bloodstream is one of dynamism, and all kind of this dynamism is existing in human body. And in this sense, the cells in lungs, for example, during breathing, the cells is under mechanical stretch because lungs expand, and the cells in blood cell wall, endocellular cells, is under shear stress. Organochip generally reproduces such organ-specific dynamism or dynamism of surrounding environment. There are several platforms available regarding organochip. The first one is single chip type. This single chip type allows us to culture organ-like cells in one chip, one organ, one chip. But its interesting point is interconnectability. The different organochip can be connected by tube, and blood mimicking culture medium can flow through the chips. So that there are several things to be addressed, but the ultimate goal of this type of chip is to create body-on-chip system in the future. The second one is culture plate type. This type of organometer chip deploy dozens of chips in one culture plate. So we can consider the biological study design more flexibly. I mean, we can have multiple concentration of multiple chemical substances and with multiple replicates. So it's very high throughput, so that's useful for the toxicological screening or some comparative studies. And third one is multi-organ chip. As name suggests, multi-organ chip allows us to culture 2 to 3 organ-like culture on one chip system. And blood mimicking culture medium flow through the organ so that organ-organ interactions can be observed with this chip. Yeah. And another point of this advantage of this type of chip is the capability of application of traditional cell culture, like 2D culture and 3D culture can be placed on a gray circle in this figure. So researchers select organometer chip based on their research purposes or experimental needs. So I'd like to introduce our case study with vascular chip for risk assessment of tobacco product. And in our case, we used vascular chip in culture plate type. This is a schematic view of our vascular chip system, and cells isolated from culture vessel wall, endocellular cells are cultured in cell culture lane, supported by collagen gel loaded in extracellular matrix lane, ECM lane, and in doing so, the cells can form tubular structure like actual human vessel. So we can add culture medium and flow through the tube, and we can also add leukocyte or immune cells to the culture medium to see the interaction between the immune cells and endocellular cells. So this is very much human relevant in vitro system, so that we expect that is a predictive in vitro system. As I said, this case study is to evaluate the heated tobacco product, and this is our proprietary heated tobacco product, Plumex. And this employs a direct heating system of tobacco leaves and resulted in the lower levels and fewer chemical constituents in the aerosol. Harmful and potential harmful constituent is reduced in the aerosol of Plumex compared to cigarette smoke. So we expect a reduced risk potential of Plumex. And in this study, we investigated the reduced risk potential on cardiovascular disease compared to cigarette smoke. So cardiovascular disease is world's biggest cause of death, and one of representative disease is artery disease. It's very much complex disease and condition, but one of the triggers is known to be oxidative stress and oxidative injury. Oxidative stress further causes inflammation of the artery, vessel, endocellular vessel, and then the endocellular cells is damaged, so that barrier function of endocellular wall is deteriorated. Barrier function, that the normally blood vessel endocellular cells has a barrier function to separate blood components like LDL and subendocellular compartment, but if the barrier function was deteriorated, the blood components can penetrate into subendocellular compartment. And also, important feature of artery disease is adhesion molecule and the leukocyte endocellular adhesion, because inflammation causes the upregulation of adhesion molecule in adhesion molecule expression, And the adhesion molecule is a protein that can capture the leukocyte in blood flow, then resulted in the leukocyte endocellular adhesion. The captured leukocyte eventually migrate into subendocellular compartment and uptake LDL, then form lipid-rich cells, namely foam cells. Foam cells is foamy and frothy appearance in microscope and is a direct cause of artery disease by its accumulation. We focused on the early stage artery disease enclosing square, and a comparative study was conducted. So we first investigated the oxidative stress in vascular on a chip by exposing Prumex aerosol or cigarette smoke. Oxidative stress was assessed by reactive oxygen species and glutathione depletion. Reactive oxygen species, ROS, is the radical molecules that attack the cellular component, and finally cell is damaged or eventually death. And glutathione, on the other hand, is the antioxidant that scavenges ROS, so that increase in ROS and depletion in glutathione is a specific feature of the oxidative stress. And we also evaluated the cell viability as an index of oxidative injury. And as you can see, cigarette smoke in red line shows an increase of significant increase of ROS and also significant decrease of glutathione and cell viability. But on the other hand, Prumex did not cause such adverse biological events, so we expect that the Prumex is not the cause of oxidative injury. Yeah, we also assessed inflammation. Inflammation is also a specific cause of artery disease. And the inflammation was assessed by observing the pro-inflammatory cytokine interleukin-1 beta or IL-1 beta by exposing Prumex aerosol or cigarette smoke to macrophages. Macrophages is immune cells that control inflammation or some biological process. And as a result, cigarette smoke induced the secretion of pro-inflammatory cytokine, but not in Prumex. So that, yeah, Prumex is not a cause of inflammation. Then we also assessed the barrier function of vascular on a chip. Barrier function of vascular on a chip was assessed by adding fluorescent dye inside a tube of endocellular cells. And if the endocellular cells was damaged, the comparable concentration. Next, we investigated the adhesion molecule. Adhesion molecule is a protein, so that we used antigen-antibody interaction with fluorescent probe. And you can see the brighter image in cigarette smoke exposed vascular on a chip, but less in Prumex exposed vascular on a chip, even at a higher concentration than cigarette smoke. So induction of adhesion molecule was also caused by Prumex aerosol. So we further investigated the monocyte adhesion on the endocellular surface. Because if adhesion molecule upregulated by cigarette smoke function properly, the monocyte can be trapped on the endocellular surface. So we assessed the monocyte adhesion. Monocyte is one of immune cells, leukocyte in blood flow. And we can add, as I said, we can add leukocyte in the blood flow culture medium and flow through the tube. So we use such approach. Then each dot presented in the right bottom panel represent monocyte trapped on the endocellular surface. And you can see a greater number of monocytes are there on the cigarette smoke exposed vascular on a chip, but less in Prumex aerosol. Overall, our result demonstrated that at least the early stage of artery disease, Prumex showed less pronounced effects. Of course, we understand that this vascular chip is perfect. I mean, that it does not purely capture the complex nature of artery disease and also physiology of vasculature. But we expect that reduced risk potential at least the early stage of artery disease. And also, we understand further study is still necessary to conclude that Prumex is reduced risk potential. For example, clinical trial and epidemiological studies can provide deeper insight into the reduced risk potential of such heated tobacco product. Maybe, yeah, I expect you may be interested in the other scientific evidence around Prumex, and we brought some of them in this conference. So please visit us at booth F2 during the coffee break just after this presentation. And our scientists are waiting for your visit and looking forward to discussing with you. Thank you. And before finishing my presentation, I'd like to thank my colleagues, Kazuhiro Ayaka, Kazushi Atsuko, all in Japan side, conducted this work. And also, I'd like to thank Mimetas. That is actually vascular chip is purchased from Mimetas, and they help us a lot. And also, I'd like to thank my boss and colleagues in JTI International. Thank you for your attention.
Thank you, Ito-san. Wow. Oh. I don't think I'm sufficiently qualified. I do have some questions, but I don't want to reveal my ignorance in front of a lot of other people. So I'll leave the people who are far more technically adept than me. And I see somebody over there.
James Sprieger from the US. Just two quick questions. The first is, what is the status of regulatory acceptance of this method? Like, for example, would the US Food and Drug Administration accept this evidence? And then second, I'm curious, what's the source of the cells? And would different chips have just as much variation in response as different humans would?
That's a good question. For the first question, actually, as far as I know, that kind of organochip system is not accepted in terms of regulatory authorities at this moment. But there are several efforts to tackle with. And second one, sorry, what's your question?
The second question was, what is the source of the cells for the chips? And would different chips have just as much variation and variability as human subjects would?
Right. Yeah, you mean the donor variation in the chips or different organ on the chip resulted in a different result?
Would have exactly the same response to the same stimulus, or would it have as much variation as the population would?
Right, yeah. Actually, there are donor variation in as far as we use primary cells isolated from the human, healthy human. Yeah, so that is an issue to be addressed. And also, different organ chip can result in a different result because the physiology is slightly different, right? Is it answer for you? No, sorry. Yeah, okay.
No, I can't. I can't see anybody else, and maybe I'll meet those who have got questions in the same place in terms of meeting up with Ito-san outside. Thank you very much indeed for that.